Welcome back, friends. This is the fourth video in the lecture series of membrane proteins, and here I am going to discuss about the next topic, which is very important. It's about the alpha helix, the alpha helical conformation of the alpha helix structures, which is found in the transmembrane proteins. So the first point here is about the transmembrane proteins. We have already discussed about the types of membrane proteins, and this transmembrane protein is one of them. So if you haven't watched the previous videos about these membrane proteins, I would suggest you please watch them and then come here. So we are only interested here about the spanning segment of the transmembrane protein. So it's about the segment which is going to span the hydrophobic environment, which is formed by the fatty acid chains. So what is that segment? It's mainly formed of alpha helix. So the spanning segment is having the conformation of alpha helix. Okay, so why this alpha helix is present in these proteins, that's the question. Okay, and we need to understand the basic knowledge behind this. So let's discuss that. First, I want to draw something here. It's about the spanning segment. Suppose it's the spanning segment, it's like this, but for the sake of understanding I would draw it like this. And our plasma membrane, there are two layers, so it's like this, and this protein segment is spanning the membrane. So there are two portions here. This one inside is the, you can say here, and there is one which is outside. This outside portion here also, it is made up of phospholipids like this, and these are the head groups. What you have to note here is that the outside portion of this protein is facing these fatty acids, okay, and these fatty acids, they contribute to the hydrophobicity of the membrane. So if we logically think, the amino acids present on this particular portion, the outside portion of this peptide part, it would be formed of what? The hydrophobic amino acids. It makes sense, isn't it? Yes. And what about the inside portion, which is away from this outside part? So it would be formed of, it would be made up of hydrophilic amino acids. So that's clear. Let's go ahead. Now this hydrophobic environment, this is very important, listen to it carefully. This hydrophobic environment, this is going to have a bad effect on the hydrophilic interior of this span segment. You simply say this is not good for the interior portion of the peptide part. So we need to save our protein. We need to protect our protein from this bad effect. So what to do now? How is that going to happen? So there is a solution for this, and that's hydrogen bonding. So hydrogen bonding is responsible for rescuing our interior portion of the spanned segment, that peptide part. So this hydrogen bonding is coming to play now. But the question here is, how is that going to happen? So let's discuss that. We know water is absent. If there is hydrophobicity, it means there is no water. That's so simple, isn't it? So there is, we can say hydrogen bonding is not going to happen. Okay, but the peptide bonds, the bonds which are formed between the amino acids, they have a tendency, and that is to form hydrogen bonds with each other. Okay, yes. Now listen to it. The amino acids which are present on the interior part of the peptide part, okay, they have polar peptide bonds, and we just discussed these polar peptide bonds, they have the tendency to form hydrogen bonds. Okay, now let's draw a diagram here. Suppose this was our protein, and just draw it like this. Okay, this is our spanning segment, and there are proteins, there are amino acids which are present like this. They are different. I'm not going to show them with different colors, it would take a lot of time. So the first amino acid which is present here, okay, it has a carboxylate group like this, okay. And there is one more amino acid here at the 5th position which is having this group. So they are polar, okay, and so they are free, we can say. They are not in bonding, so they are free, and these groups, they are vulnerable to the bad effect of that hydrophobic nature, okay. So what they do, they make bond, and this bond is known as here H bond, that is hydrogen bond. Clear? So hydrogen bond has been formed, and this kind of bonding is seen in what? Alpha helix conformation or alpha helical conformation. So that's what about alpha helical conformation in membrane proteins. Now the thing here is that if this bonding is occurring, it means these groups are not free, so they are in bonding. So in this way our protein is shielded from inside from the effect of the hydrophobic environment. Now there is an important point, that is this hydrogen bonding is maximized. So we are here for hydrogen bonding. If there is more hydrogen bonding, it means there is more shielding of protein. That's good for us, okay. So this hydrogen bonding is going to maximize, it is going to increase if the polypeptide chain, if the polypeptide chain which is present outside initially, and now it's going to enter the cell membrane, and upon entering, if it's assume or if it's opt for the alpha helical conformation, it is directly going to increase the hydrogen bonding. So this is how majority of the membrane spanning segments traverse the bilayer. That is why we say that the transmembrane proteins, they are having alpha helix in them.